We are like rivers. A river flows. Water is never same in the river. It flows away. But the river is same always. Same happens in our body. The cells renew again and again. Old cells die, new cells are produced, and they replace the old one. This process is like flow of a river. It continues. The cells are not same always. After each 7 years, most of the cells in our body are replaced by new one. But we are the same, like a river. In the river, water is never the same, but river remains the same.
Our body is a vibrant community of nearly 220 different types of cells. There are populations of these 220 different types of cells in our body. Apart from that, our body has almost 8 to 10 times the number of bacteria housed within it. Hello friends, this is Lakshmi Raman Ozha from SAMARTH Biology Tutorials LATUR, and you are watching SBTL webcasts. In the last lecture, we discussed about one of the features of living organisms, that is metabolism. It was a very brief discussion in fact, and this time we are going to talk about the next feature found in living organisms, and that is cellular organization. We are going to talk about two things today. The first thing that we are going to discuss is the differentiation of a single cell, what we call as zygote, into various different types of cells, almost 220 different types of cells present in our body. So we are going to talk about this differentiation of cell. And the second thing we are going to discuss is internal organization of cells. For example, prokaryotes means bacteria. Few of them live in the form of colonies. So how the structures of cells, they differ in various different parts of that entire structure. And we are going to talk about eukaryotic cell and its internal compartmentalization as well. So let us dive in.
Our journey in this living world takes place almost 9 months to initiate. This is the period in which a single cell grows eventually through many transformations into a complete physical body. This is shaped during a gestation period of almost 9 months and 9 days. Then our body shapes completely. It is ready to enter the living world. This entire journey begins with a single cell, and that cell is called zygote, that is formed by union of a male gamete with a female gamete. The fabric of human body is woven in a span of 38 weeks, 261 to 267 days, and this journey of a single blob of cell called zygote into a fully developed human form takes place in this period. This period is called the gestation period. In lower animals like kangaroos, few earlier mammals like kangaroos, there is a pouch on abdomen that is called marsupium, and that is the reason why these animals get their name marsupials. When the newborn is not completely developed, fully developed, the further development takes place in this marsupium. In humans, the complete development of a zygote into a full developed form takes about 38 weeks, a span of nearly 261 to 267 days, or you may say 9 months. After the fertilization, on the next day, divisions starts. These divisions taking place in the zygote, they are called cleavages. That results into 2-celled condition, then 4-celled condition, then 8-celled, 16-celled, and so on. All these cells are grouped into a single mass, a single lump, a single bunch. On the 4th day, this looks like a fruit of mulberry. The scientific name of mulberry is Morus alba. Morus is the name of its genus. Embryo looks like a fruit of mulberry, and hence it is called morula. All these cells of morula are totipotent cells, means each of these cells is capable of giving rise to entire living organism. If we separate any of these cells, then we can grow a complete new clone of that organism from it. Morula further divides. In this journey, the first 4 weeks are important. In these 4 weeks, a span of 1 month, the invisible embryo grows up to 6 millimeters. It has developed germ layers into it during this journey of 4 weeks, 1 month. These germ layers are highly specific. They give rise to highly specific systems in our body. And here the process of differentiation begins. Our body has a supportive framework called skeleton. This skeleton is composed of bones, cartilages, and joints. Nearly 206 bones, they make this structural framework. But apart from that, bones are the places where some cells which can produce our blood are located. Bones in our limbs are long bones. These bones, they have an outer cortex and inner medulla. The cortex is made of compact bone that is filled with minerals like calcium and phosphorus. Nearly 99% of calcium in our body is located in this region called compact bone, that is the outer part of each bone. Just below that, there is something what we call as spongy bones, and in this spongy bone there is a tissue called red bone marrow. In the bone marrow, there are some cells which can grow into bone forming cells called osteoblasts, bone destroying cells called osteoclasts. For example, when our body needs calcium, then osteoclasts, they break the tissue of bone little bit, and the calcium that is deposited there becomes available. Bone forming cells are called osteoblasts. Bone destroying cells are called osteoclasts. And couple of more cells are produced there, spatially the cells in our blood. Our blood has variety of different cells. Most of them are red blood cells or erythrocytes. They have an important function to transport oxygen towards the cell in our body. Formation of blood cells is called hemopoiesis, and that takes place from this tissue known as bone marrow. These cells, which are capable to give rise to various different types of cells like osteoblasts, osteoclasts, bone cells, their precursor cells, their earlier cells, they are known as their stem cells. So the process of differentiation, it becomes highly organized eventually. Let us take example of plants also. Leaf is the site of photosynthesis. If we take section of leaves, we can see different types of cells there. The upper surface is upper epidermis, the lower surface is made of lower epidermal cells, and in between them there are cells in which the process of photosynthesis takes place. These cells are called mesophyll cells. All these cells are differentiated. Now, when a scientist takes a section of these leaves, he cuts these leaves into a very small part, then this injury results into de-differentiation of these cells. These cells, they lose their specificity and they become totipotent again. These cells can be grown into new plantlets. This process is called tissue culture. Just like a bungalow which has many different rooms, eukaryotic cells are compartmentalized. Each compartment has its own unique environment. Enzymes in that compartment work in that specific environment only. They need a specific pH, a specific solute concentration in the surrounding fluid. While bacteria are cells with no compartmentalization, it is just like a single room. But some of the bacteria, they evolved over time and they lived in colonial forms. One of them is blue-green alga, or that is cyanobacteria. They are the first photosynthetic autotrophs on these planets. These bacteria make filaments, but differentiation can be seen here also. Usual filaments of these cyanobacteria, they have green photosynthetic cells, but they have one more role. They fix the nitrogen, and hence they need a compartment, a differentiated cell that can work in non-photosynthetic condition, because oxygen inhibits the process of nitrogen fixation, and they develop some cells. They are called heterocysts. This is how differentiation can even be noted in colonial prokaryotes. So, we discussed two things in the cellular organization. One is how the cells differentiate from a non-specific form into a highly specific form. And the second thing we discussed was organization within a cell. Eukaryotic cells are highly compartmentalized. On the contrary, if you see a prokaryotic cell, then that does not have any compartment. However, you can note that bacteria developed colonization later. They lived in groups. Few of them formed filamentous colonies, just like cyanobacteria. And there also the process of differentiation can be seen. You can see the process of differentiation even in those colonized bacteria. There develop some cells like heterocysts which lack photosynthetic apparatus in them, because photosynthesis releases oxygen, and oxygen filled environment does not allow nitrogen fixation, the functioning of enzyme nitrogenase. So that is even a form of differentiation we can see in prokaryotes. Friends, thank you for listening me so kindly. This episode was a bit long, and see you again in the next part. Thank you again. Thank you.